on Canvas, right? Since obviously we're done, right? So next week you're not going to come in and take a quiz. Next week you're going to come in and take a test, right? So to help you out with today's experiment, what I did was put a quiz, practice quiz on Canvas for you guys. So make sure you study the material, right? Take the quiz, see how you did. It'll tell you the answers, right? And help you know if you really got it. Okay? So in addition, we're going to do final exam review, which you have a little thing for that in front of you that we're going to go over in a moment. And then your unknown report, of course, is an excellent, excellent guide for the final exam. So I'm actually going to start with that one first. So this guy, right? So as usual, grading it at Jazz Fest this past weekend. I only got to go on Saturday though, but it takes a long time to grade these, right? I really take my time, as you can see. I write all over it, and I think this is the one from last semester. Even it never changes. What people miss. So, some insight would be helpful, you guys, if you could help me as to why you guys don't do these things. I don't know. But one of the things that came up again is this page. Your negative slide. What should your negative slide look like? The background should be colored, right? You can do this with a pencil. It didn't have to be like that purplish gray color, right? But you should have had a colored background here. You should have had clear cells. You should have said that, right, in your description. Right? So that's really important. You're going to see a negative stain again on the test, right? You need to be able to know what you're looking at. Right? The other thing is capsule again. Not properly describing what you're seeing. It should be dark background, colored cell, clear halo. Those parts should have been labeled. Made you guys do it on the midterm. Right? I don't know why it didn't show up on the lab report. It says it right there, right? Label cell and capsule if present. Right? So some of you guys lost points on that. Really easy points, mind you. Right? So the genus and species names should always be italicized or underlined. I don't really take off for that, like on the last page, because I have a line for you guys to write it on. And I'm really bad about that too, because I type everything. But I just want to, emphasize that to you guys. You'll notice that they should be, in that table, they should be italicized, right? The actual names. I'm not as big stickler on that one as some other professors are. But really should keep that in mind, right? As you guys are writing this genus and species names of the organisms on the final exam, you should underline them, right? We only had two choices, right? Cocci, spheres, or bacillus, which is rod-shaped. And again, that's where sometimes, italicizing is going to help out, because are you talking about the genus bacillus, or are you talking about the shape bacillus? I was slightly nervous. I thought it was another noise. I was like, for real? All right. The other thing with Graham, right, you should capitalize that. That's actually a guy's name that came up with this staining procedure. If you don't capitalize it, again, sometimes it could be confused with gram as in the unit of measurement, right? That's a weight, a unit of mass. I know, it sounds like... Okay, so gram staining, right, probably the most valuable of the stains that we do, because it really divides up the possibilities in half, right? About half of all the own bacteria are either gram-positive or gram-negative. And as you'll see by our chart, too, even what we had to choose from in this class, right? Half of them were gram-negative and about half were gram-positive. So remember the structures and why it is, right? And remember, gram-positive are going to be what color? Purple, right? Because they retain the crystal violet iodine because they have a thick peptidoglycan layer. Where gram-negative's thin peptidoglycan layer, they don't retain the crystal violet iodine. Their outer membrane actually gets destroyed, and then they get counterstained and they're pink. So even though, we did controls on that slide. That was the slide with the 3 circles, right? Even with doing that, sometimes the reliability, this is a difficult technique for some people, especially beginning students. And you guys are lucky you don't have to do it as part of your practical exam, and the future students will have to actually do a gram stain for their practical exam. So all of these different stains really could help us with morphology, as well as the negative stain I find is probably one of the easiest one for students to see arrangement. And that's one of the reasons why I make you guys do that one, because that's pretty much all it tells you, right, is shape and arrangement is a negative stain. Unlike all the rest of these, right, gram stain is going to tell you positive or negative, like we said, purple or pink. For a capsule stain, it's going to tell us if you have a capsule or not, and you can clearly see one here. For endospore stain, it's going to let us know if there's an endospore. What would we say is the arrangement here? Again, this is really easily seen arrangement. Rod-shaped, and they're in pairs, so we would say this is diplobacillus, right? Okay, these are singular. These are forming chains of rods, so these are streptobacillus, right? That'd be streptobacillus. Clustering of cocci, staphylococcus, right? And this, remember the acid fast, a lot of times they tend to clump together, but they are bacilli. That's why sometimes it's AFB, right? Acid fast bacilli, but sometimes it's hard to see that they're actually rods. They're going to be that fuchsia purplish color, and your non-acid fast would be another color. Most of the organisms we dealt with were actually encapsulated. Most of you guys, actually, in fact, all of you guys should draw in a capsule and labeled it in a cell. Motility. Most commonly, motility by bacteria is by flagella, right? We didn't actually do a flagella stain, but we did 2 tests, right, that would tell us motility. We did the motility stab, which you can very easily see. If they don't move, it's negative. If they moved out, they were positive. There was, Ashley had the one organism, right, that was a little too big to move, so we looked at it on the microscope and we're like, oh, wow, look at that, right? So we actually did a wet mount to confirm for hers. And I think we may have done that for one other person. Maybe Caitlin, who's not here, I think. I don't know. I don't remember. Maybe I'm thinking of my other class. There's one of them that sometimes in the motility test doesn't look very modal. And so we usually look at it swimming under the microscope. So our FTM tubes, this is another one where people don't do what the instructions say, right? You were supposed to draw the oxygen zone and the non-oxygen zone on your tubes and label it. So I don't know, maybe I'll put like these little markers, right, on the tubes or something. Maybe I can try and, I don't know what to do. Get you guys to fill it in. I drew drawings on the board, didn't I? I don't know what else to do. Okay. So it's important, right, because we have an oxygen zone and a non-oxygen zone, and we really need to know where the growth is occurring, right? So for us at Delgado, right, we don't mess with the anaerobes because it's too hard to keep oxygen out of this lab and keep us alive. Microaerophiles are also tough to keep happy because we got to keep a small level of oxygen, and that would kill us too. So not interested in everyone passing out in labs, so we don't mess with those guys because we need the special jars and everything, right, for them. And special equipment. So we really just had these 3 choices, right? And so strict aerobes would only grow in that oxygen layer, right? And that was because they were utilizing oxygen. Then we had facultative anaerobes and aerotolerant anaerobes. And sometimes, especially depending on how old the culture is, it'll be tough to determine one from the other. One of the ways you can determine the difference is you'll notice the facultatives have more growth at the top than at the bottom. More growth in the oxygen zone. Why is that? More growth. They can grow in either environment. So do they need oxygen to grow? No, they can grow down here without it. But what's the difference between having oxygen and not in their growth pattern? They can do aerobic respiration and produce a lot of energy, so you get a lot of growth, right? But without oxygen, they can anaerobically usually ferment, right? This guy, on the other hand, aerotolerant, which means it can tolerate oxygen, right? But does it use it? No, it's growing anaerobically, it's fermenting. That's why its growth pattern is uniform throughout the tube, right? Because even though oxygen's here, it's not using it, right? It's fermenting throughout the entire tube. The difference is, unlike our strict anaerobes, oxygen's not going to kill it. So this relates to another test as it relates to whether they are using an electron transport chain and using aerobic respiration, which for these guys, it's only 2 of them, right? Aerobes, clearly, electron transport chain, oxygen, final electron acceptor, right? Same thing for your facultative anaerobe. When oxygen's there, using electron transport chain, oxygen, final electron acceptor. Is this guy doing that? Is he doing aerobic respiration? No, right? We just said he's fermenting, right? Not using the electron transport chain. So what other tests did we do that would actually help you distinguish between an aerotolerant and a facultative anaerobe? Christy knows. Someone over here said it. Well, catalase, right? Hers doesn't have catalase, right? Right? Because hers is the aerotolerant. It's not using aerobic respiration. So it doesn't need catalase. Right? Because when we look at catalase, who has it and why? And I'm going to jump ahead. Right? So it detoxifies hydrogen peroxide. Well, where does hydrogen peroxide come from? It comes from having an electron transport chain where electrons are jumping off and jumping onto oxygen and forming hydrogen peroxide. Now, her organism has another enzyme we didn't test for, which actually will generate small amounts of hydrogen peroxide when superoxide radicals are present. That's superoxide demutase, right? But doesn't need catalase because it's not generating a whole bunch of this, right? Because it's not using the electron transport chain. Still with me? So why do we need this, right? Because we're aerobic, right? Electron transport chain, oxygen here at the end forms water. So we can break down hydrogen peroxide into water and gaseous oxygen, which is why it bubbles, right? In the presence of catalase. It breaks that down. Now this is the one test too where we can have specificity issues too if you don't perform it correctly, right? If you're doing the slide test, remember you had to do what? Because what else other than light, we keep it in a dark bottle because light will break hydrogen peroxide down. What else would cause that reaction that we would normally use to put bacteria on a slide? The loops are what? Metal. The metal could catalyze that reaction. So you had to put the bacteria first, then add the hydrogen peroxide. Because if you put the hydrogen peroxide with the metal loop in the bacteria, what's causing it to bubble? Loop or bacteria? Don't know. Right? So that specificity issue can be very easily fixed by following procedure. Right? Something me and my son learned with his science project this week. We didn't follow the instructions and we screwed it up the first time. So we put at the end, what interesting thing did you learn? It's really important to follow instructions. It was all good. We were doing kitchen science, so we just made 2 lava lamps. You know, water, oil, and fizzy antacid tablets. Lots of fun. Water, food coloring, then oil. Right? Don't add the food coloring after the oil, because then you have to wait for it to go down to the water. So we had to wait. That, and I didn't have the fizzy tablets. I tried to use the Tums. Doesn't work. Right? It says fizzy for a reason. You want bubbles. You want the bicarbonate, right? Just like when they make the volcanoes, where that's vinegar and baking soda, that chemical reaction, it's, again, bubbles, because in that case, it's carbon dioxide gas. Bubbles up. Right? But it's all about making bubbles, right? It's fun. Love to make bubbles. Okay. Any questions on this? All right, I'm going to rewind to phenol red. So, what can we detect with phenol red test? Well, what is phenol red? It's a pH indicator. What color does it turn when acids are produced? Yellow. All together now. Yellow, yellow, yellow. So we can detect acid from sugar fermentation, right? What's another product we can actually detect in this test because it has this little tube in there that a lot of people neglected to draw on their gas, right? It gets trapped in the little Durham tube and displaces the media, and we see bubbles, right? It could be hydrogen or carbon dioxide gas or a combination of both. We don't know. Depends on the organism, even depends on the pH at the time that the organism is growing, which of course changes. What else can we detect with the phenol red tubes? Is sugar fermentation the only option? No, they're going to turn, and of course Shawanda walked out because she said pink. What does pink mean though? It became basic, right? The media became basic. Which means the organisms did what? Deaminated the protein. When they eat the protein, they produce ammonia as a byproduct. Right? Turns it pink. Some organisms ate the protein really fast, right, Suda? And then started eating the protein, so then you went yellow, right, and started getting orange, and if you let it go, it would have ended up being pink. Citrate test. What does your organism have to be positive for this test? It has to have a protein in its membrane we call citrate permease. So that what comes into the cell? Citrate. Right? So if citrate comes in, they also need, that's a carbon source, right? They also need a nitrogen source. So there's one nitrogen source in that media. And the reason for that is when they use it, again, we're going to get a pH change, right? And again, it's going to be basic, right? So they're going to, when the ammonium is consumed, right, we're going to get a pH change. In this case, bromenthal blue turns blue, right? That's a positive result. What happens if your organism doesn't have citrate permease, so it can't eat the citrate? It dies. What color is your tube going to be? Green, right? Because if it's dead, is it producing basic products, alkaline products? No, right? So growth is another indicator, though, right? But remember I told you guys not to look at that? Because the problem is maybe you put too much bacteria to begin with and you might think you're seeing growth, but it's just a heavy inoculum. And so you'll notice that even in the procedure, sometimes they say use a needle instead of a loop so you don't put too much bacteria to begin with. Right? Which I didn't have you guys do because I usually have the opposite problem sometimes with you guys. You don't put enough. Then we have to do it again. That's why I just say, trust me, if it's positive, it turned blue. Right? If it's really growth, at this point when we were looking at them, it should have been blue. Alright, so we just talked about catalase. Let's go to methyl red. So methyl red, the MRVP tubes had glucose in it because, again, we're looking for fermentation. In the case of methyl red, what type of fermentation are we looking for? What do we call this? Well, methyl red is what? What's methyl red? pH indicator, right? That stays red at 4.4. It's a really low pH, right? That's really acidic. So this one is called mixed acid, right? Lots of acid, right? Lots of different acids. Usually this organism is produced that make it very acidic. So again, red is positive, and it means they produced a lot of different acids. Very acidic media. So there were some questions that I actually wrote the answers to, but did not take points off if you didn't get on the methyl red test. And it related to the phenol red test. So phenol red, right, turns yellow in acidic conditions that are 6.8 or below. So the question is, the first question is, if an organism is methyl red positive, which means that it's 4.4 or lower, right, that's the pH, when it ferments glucose, in the phenol red broth, will it be positive or negative? Should it turn yellow? Yes, right? Because 4.4 is very low, right? So if it's methyl red positive, it should also be phenol red positive, right? Because phenol red, just a little acidic, right? 6.8, it's going to turn yellow, right? Where it's going to be very acidic for the methyl red. So is the reverse true? If it is positive for phenol red, right? If it's yellow for fermentation, will it be methyl red positive? Not necessarily, right? Because you only have to be a little acidic to be yellow in phenol red. You've got to be very acidic, right, for methyl red. Does that make sense to you guys? That's what we were trying to get at there. For you guys to realize that there's two different pH ranges, right, that we're looking at. Two different levels of acidity those two tests are looking at. Did I miss, what's the third question? I think there's three of them, huh? Right, we answered that. So let's see. So can a methyl red negative organism cause the pH of the media to drop from its initial value? So think about this. The media is probably around neutral. Especially if you had one that in phenol red, right, it turned yellow. And then you do the methyl red test and it comes out negative. How is that possible? Well, because it's not low enough, right? It's got to be 4.4 or lower to be methyl red positive for mixed acid. So yeah, it's going to lower the pH, but it has to lower it enough. You understand? It's an important point. Distinction between these two tests. Okay. So Voges-Proskauer is the only test that we do that detects what type of fermentation product we haven't looked at yet. So fermentation is acid, alcohol, right? And gas. This is the alcohol. This is 2,3-butanol. But did we actually test to see if your organism had 2,3-butanol in its tube? No, what did we test for the presence of? It was an intermediate in this pathway. Acetonin, right? Acetonin combined with the reagents of our test, right? Alpha-nithapanol, potassium hydroxide, and oxygen when we flicked our tubes, combine to form a molecule that produces, again, a red color. Only red is positive. So we didn't actually test for the presence of 2,3-butanol, but acetonin is an intermediate, right, to the production of that alcohol by the fermentation of glucose. Okay, so selective media, right? The plates contained, first and foremost, were selective, which means that we selected for particular groups to grow. So if your organism didn't grow, do we know anything about the differential component of these media? No, couldn't be determined, right? You don't know if it's hemolytic or ferments lactose or ferments mannitol. It didn't grow. Notice on your lab report it says, could not be determined because no growth for the second result. That's important. So I had sent out a message for everybody on an example of all the possible results you could get for mannitol and how you would interpret that. I'm not going to bore you guys because I already see you're tired with that. So I would suggest looking back over that announcement I sent you guys and the type of write-ups I was looking for. So remember, so for mannitol salt plates, right, only salt-tolerant organisms grew. And we were using this to help us with identification of staph, but also some bacilli will grow, right? And if it does grow, we know it's salt-tolerant, right? And again, this is another thing where people left it out, and it's very important. It isn't the fact that these plates just have salt. It's a high concentration of salt, right? My shorthand is an up arrow concentration, right? A high concentration of salt. Very important. And then, if it was salt tolerant, we asked an additional question. Does it ferment mannitol? And we knew this if the plate turned what? Yellow, because what's the pH indicator in this plate again? What turns yellow in acidic conditions? Phenol red. Okay. So for the EMB plates, it's selective for gram negative. So you should have only had growth if you had a gram negative, right? If you had no growth, that tells you and confirms you had a positive, right? What kills the gram negatives? I mean the gram positives. What in this media is inhibitory? The eosin and the methylene blue, the two dyes. So if your organism survives, we can ask that next question. Does it ferment lactose, right? If it ferments lactose, it's going to produce acids that interact with the dyes. Depending on how much acid, you're going to get different colors, right? Which is our very strong fermenter, makes the prettiest color on this plate. E. coli. That metallic green sheen, that's a very vigorous lactose fermenter. This is moderate, right? Where you have the pink and the purple colors. When you don't see any pink, purple, black, or metallic green, right? When it pretty much blends in with the plate, that's a non-lactose fermenter, right? The Columbia CNA plates, the CNA stands for what? Colistin and nelidiscic acid. Who's that going to kill? It's a selective agent, right? What do antibiotics kill? Bacteria, right? In this case, though, a particular group of bacteria, gram negatives. Gram negatives die, right? So you only got this plate, actually, if you had a gram positive, because only yours was going to grow. Then, once it grew, we could ask the question, is it hemolytic? So that means some of you guys that have gram negatives, your organisms aren't hemolytic? No, it just means we couldn't use these plates to figure it out because the antibiotic killed your organism, right? We'd have to use plates without the antibiotic, with the red blood cells all by themselves, right? So, beta hemolysis, right? We could help confirm Staph aureus because it'll completely destroy, right? Beta, bad, complete destruction of red blood cells. And here's one of the bacilli. It starts to die off, so you don't see much growth, but you can definitely see that complete clearing of the red blood cells. Alpha is incomplete, right? Little holes in it, so then you get the greenish discolorization that happens to those red blood cells. With gamma, is there any destruction to the red blood cells? No. Gamma hemolysis actually means no hemolysis. And actually, that was the easiest question on your guys' lab report, right? You literally could have wrote just no. So justification of inconsistent results is important, right? So it's okay if you didn't get what you were supposed to get, right? If it didn't all match up. That's what the last page was for, to talk about that. You know, why was this one result or this other result off from what was expected? What could have gone wrong, right? Only a few people got dinged on that. All right, so let's practice, right? So for our exam, what should we look at? Well, one, you should look at your syllabus, right? There's a reason why we give it to you. We even print it out for you in this class. We spend a lot of time putting that together. It's very well detailed, right? Actually read some of those objectives that we have written in there, right? What we want you to be able to know and do from this course, right? Always, as always, homework and quizzes, excellent guides, right, for studying for the final exam. And again, remember, terminology needs to be spelled correctly. Go back over all your homeworks, make sure you know all your terms and you know how to spell them. I know. Your graded unknown report, which I gave you today, is an excellent study tool, right? Because all of that is on the final exam. You will see all of those tests again, right? And have to be able to interpret them or ask questions or answer questions related to them. In PowerPoint lectures, focus in on those scientific method, right? See if you can remember. Why did we do this? How did we do it? What do the results tell me, right, about my organism, right? That's why those write-ups are really important, right? Kind of summarizes it and puts it all together. Create your own tables of information, right? Summarize stuff. Right? Sometimes writing out in table form helps people. Covers all exercises after midterm, right? Most of them were used to help identify your unknown. But we had some that were all by themselves. Standalone experiments like the one we did today. So all of this stuff, right, related to your unknown. Notice this one we just discussed, and I can guarantee you I'm not going to ask you any questions about it on the test. But the anaerobic jar, remember we discussed it, but we didn't do it. That one, right, you still need to know. The ones that are by themselves, right, is the standard plate count. Make sure you go over that. Antibiotic sensitivity, remember. P-glow, which we did last week, right? Transformation. And ELISA that we did today. So you should understand the process of unknown identification, right? All the steps that we used, huh? Mm-hmm. Oh, why not? Oh, yeah. Maybe this one is not on there. These aren't on here? On there? Oh, because right, because 9 isn't there. Yeah. Yeah, so probably anaerobic jar might not be on that list too, right? Yeah. Yeah. It's in the standalone part? Okay, yeah. But this PowerPoint I posted for you too. Okay. So you should be able to go through this process, right? Be able to evaluate. You're going to do the same thing on the exam. There's going to be slides. You got to tell me, is it gram-positive? Is it gram-negative? Is it acid-fast? Does it have a spore? Does it have a capsule? All of that, right? Negative stain, maybe, because that one's good for arrangement. Tell me the shape and arrangement, right? Or the term we use for the shape and arrangement, right? So I might put some staphylococci or streptobacillus up there for you, right? Yeah. Okay. So we got to do all that fun stuff, right? And then given data, right? I'll give you, right? Data, like just like what you guys did right here, and you got to figure out what it is. Of course, we're going to give you the table, right, of known results. You're not memorizing that, right? Although I pretty much almost have the whole thing memorized at this point, except for the VP and the methyl red. Those don't really stay in my head. But all the rest, I could probably rattle them off to you guys. I've been teaching this long enough that I've memorized. Scary, but true. Okay, you're not memorizing them. You don't have any need to, and I've accidentally memorized them. It's repetitive, right? When you do something over and over again, you remember it. So let's see, what did you guys get for this one? And this one should glare out at you. There's one result here that for this class really keys in one answer. Yeah, this is Mycobacterium smegmatis. And what test result screamed that at you? Acid-fast. Only one, y'all. In the future, I'll probably have 2 mycobacteria. But yeah, it'll still narrow it down to 2, right? If it's acid-fast, you're probably looking at mycobacteria. right? And then, of course, the other test results confirm that. So what about this next one? This one you could narrow it down to 2, right? The 2 what? The 2 staph. Right? So then, so here we are, gram positive, so that gets rid of half of them. Right? Phenol red, got rid of those 2. Citrate and motility narrowed it down to these 3. And then the catalase eliminated the Enterococcus and left us with the 2 staphs, which pretty much match up except for VP. And again, would you really want to rely on just one test? No. And we don't. So what do? We do plate media, right? Which one would help you with Staph aureus versus Staph epi?
Mannitol salt?
Mannitol salt, because on mannitol salt, Staph aureus is going to be yellow, right? That's the one that turns the plate yellow. That's why we use that media. And this is one you do need to know, right? It's one of your questions on your unknown report, right? What are we looking for when we use mannitol salt? Usually looking for Staph aureus. We're looking for that yellow. Where Staph epi is not going to change color. Is there another plate we could use to help us out? Tyler?
I was thinking about the blood agar.
Right. No, you're right. You're right. The blood agar. Because we also know from your lab report, and I don't want you guys memorizing these, but Staph aureus, Bacillus cereus, and Bacillus subtilis are all beta hemolytic. Enterococcus sometimes is alpha. Right? So that plate would additionally help out. And again, so now we have, 3 things that should come out different for the 2 different staphs. Okay, this one was an answer, right? Which one? Yes, Serratium recescens. I think I forget. I don't know if I never did post the guide for you guys to learn how to pronounce these. I need to do that. So again, we could cut it in half by doing gram, right? So half of them are gone. We're down to the negatives. The methyl red got rid of 2 of them. The citrate test got rid of 3 of them. We're down to Klebsiella and Serracea. And the phenol red really helped you tell the difference between the 2. And in fact, this one was one where if you it'll start eating the protein too, so sometimes I get that little orange layer on this one. So a weak K also for this one. So next one, what are possibilities? Right, so this is where we're down to the 2 bacilli, right? So this is the one, I'll go quickly. Right, we're down to the 2 bacilli. And again, one test is different for these guys. Which test? Yeah, it's the VP test. It's the only thing different on these 2. So would you really want to rely on just that one test? No, so we would do plate media, right? And the good news with this one, and one of the reasons why we do it, right, is the Bacillus subtilis, as I said, would be beta hemolytic, where Bacillus megaterium is not hemolytic at all. Again, don't want you guys memorizing those. What ones are beta hemolytic versus alpha? That's right. But just reinforce why did we do more tests? That's why, right? You don't want to just rely on one result. So, first half of the course was all basically anatomy, right? And now we've been focusing on physiology. In the rest of this PowerPoint, which I'm not going to go over, again, we're not revisiting these, but don't forget, right? Why do we use microscopes? They create resolution and contrast and magnification. The good news, not on the final, right? But understanding and interpreting stains, right? So making tables like this one, seeing if you can fill it in, right, are helpful. This is a good one for going back over standard plate count. And yes, the formula you do need to know right there, right? And what parts come from where to fill it in, right? So those three things, right? Figuring out individual tube dilutions. You will have a problem on the final, a worksheet, right? Fill in just like this, right? Or you got to have all these things, right? And you put it all together to give you your answer. Kirby-Bowers, super easy, right? Antibiotic sensitivity, right? Interpreting those results. Again, I'll give you the table, right? I'm not making you memorize how big does the zone have to be for penicillin or ampicillin, right? It'll be very much like your quiz, right, on this. And then all the different tests. I'm trying to think. And bacterial transformation. And then, like I said, for ELISA, do that practice quiz I gave for you guys. But you also have this PowerPoint. Help you go through and review, right? And remember all the finer points.